Welcome to the Global Virus Network's Forefront of Virology webinar series. I'm Dr. Maggie Bartlett, the Program Director for the GVN and your moderator today. We are so pleased to have Dr. Yi-Hao Chan with us to present on genetics as a compass for life-threatening viral encephalitis, one of my favorite topics. Dr. Chan is an A-star scholar and did their PhD on alphavirus immune pathogenesis. They did their postdoc at Rockefeller University, focused on neuro-intrinsic antiviral factors in herpes simplex encephalitis and SARS-CoV-2 encephalitis. And now Dr. Chan leads a team at A-Star Infectious Disease Lab studying genetics of immunology and severe viral diseases. And I am so pleased to hand it over to Dr. Chan to enlighten us on this fascinating topic.
Great. Thanks a lot, Maggie. First, let me share my screen and put it on full screen. Okay, I hope everybody can see my screen. Thanks for the kind introductions. My name is Yi-Hao, a principal investigator at A-Star Infectious Diseases Labs here in Singapore. Today, I'm going to present on my work, which is a majority actually part of my postdoc work in Jean-Laurent Casanova's lab in Rockefeller University. And the title of today's talk is Genetics as a Compass for Life-Threatening Viral Encephalitis. So before I begin, I'll dive deep into the topic. I'd first like to introduce to everyone here the idea of the infection enigma. This is actually a clinical observation of the immense inter-individual clinical variability that we see in humans or populations being infected by the same common benign pathogens. And this pathogen can be of any microbe, be it viruses, bacteria, fungi, and so on. I think the most, the popular, well-known case will be the COVID-19 pandemic, where, a huge percentage of us actually got infected during the pandemic, the 3 to 4 years of a pandemic with SARS-CoV-2. But fortunately for us, there were, the invention of RNA vaccines, the rollout of RNA vaccines that protected us. But what is more important is the observation that majority of us actually, develop only mild symptoms, if any at all. Majority of us, I believe, remained asymptomatic. But unfortunately, there are some rare few incidences of severe COVID-19 pneumonia requiring intubation. And through pioneering works of inborn errors of immunity in Casanova's lab, as well as many other labs across the globe, we found that genetic deficiencies to type 1 interferon pathway, as well as autoantibodies that neutralizes type 1 interferons, can be predisposition for individuals to severe life-threatening COVID-19. So here in my lab, we believe that by cracking the infection enigma, we can revolutionize our understanding of the human biology as well as medicine. As many of you already know through the introduction by Maggie, I did my postdoctoral training in Dr. Jean-Laurent Casanova's lab and also under the supervision of Professor Shen-Ning Zhang, where we worked in Rockefeller University to find genetic basis of childhood herpes simplex encephalitis. Of course, the causal pathogen of herpes simplex encephalitis is herpes simplex virus 1, which is a very common benign pathogen. Majority of us, I would say around 70%, have already been exposed or been infected by herpes simplex virus 1. But fortunately for us, 99% of us actually get, just asymptomatic during the acute phase of infection, or even just mild symptoms, like, for example, cold sores at the mucocutaneous areas, like the lips, or even just low-grade fever. But unfortunately, there is a rare few incidences of life-threatening herpes simplex encephalitis in infected individuals with select genetic deficiencies. Through research and pioneering work by Dr. Jean-Laurent Casanova and his team, as well as many research groups across the globe, we found that genetic deficiencies, primarily in the TLR3 type 1 interferon pathway, can predispose individuals to herpes simplex encephalitis. And this does not exclude genetic deficiencies outside of the DR3 type 1 interferon pathway, because we did find genetic deficiencies independent of the type 1 interferon pathways, including SNOR31 deficiency, DBR1 deficiency, RIPK3 deficiency, to name a few. So but because of this work, we have genetic explanation or etiologies for 10% of the HSE cases. So our hypothesis at this point is that there are other genetic etiologies that could explain childhood herpes simplex encephalitis. So using a genetic analysis approach on our in-house control cohort of more than 24,000 whole genome or whole exome sequencing data, as well as our in-house herpes simplex encephalitis cohort consisting of 319 patients with herpes simplex encephalitis, we did a genetic analysis to find new genetic variants or mutations that could predispose individuals to herpes simplex encephalitis. We excluded the patients with HSE with already known genetic etiologies, and with the remaining 290 patients, we went through an optimized genetic variant selection pipeline, and we found two unrelated patients with homozygous mutations in TMF1, which we label as patient 1 as patient 2 hereafter. Patient 1 is of a North African origin, who was diagnosed with herpes simplex encephalitis at 2.5 years. She was born of non-consanguineous parents, and through whole exome sequencing, she was found to have a missense mutation at the 44th amino acid position of TMF1 near the extracellular domain of TMF1, from a proline to a alanine. While in patient 2, he was diagnosed of herpes simplex encephalitis at 4.5 years old, and he was born of consanguineous parents. And through whole exome sequencing, he was found to have essential splice mutation, 2 nucleotides before the exon 10 acceptor site. And this can result in many different isoforms of TMF1 being formed during protein expression. Through search in the NOMAD database, we also found 13 other individuals with homozygous variants in TMF1 across the TMF1 gene, 8 different mutations, including 2 individuals with the same mutation P44A as patient 1. So looking at the TMF1 population genetics, from 2 patients labeled in red, we can see that through the CAT-MEF graph, they have a high CAT score, signifying that they are potentially deleterious. And looking at the MEF, they are rare, less than 1% of the general population. And looking at the CONES score, they are the genetic variants of TMF1 are predicted to be inherited by autosomal recessive fashion. And looking at the NOMAD database, we can see again 2 other individuals with homozygous P44A in TMF1, while there were no other individuals with the essential splice mutation like P2 existing in homozygosity. There were also no homozygous variants in the Bravo as well as GME variant databases. At this point of the study, we actually do not know, what is the actual function of TMF1 other than a few association studies of TMF1 with prostate cancer. So to start off, we just wanted to find which tissues or which cell types express TMF1 endogenously at the highest level. And through qPCR at different RNA samples from different tissues and cells, we found that TMF1 were expressed in the highest level, especially in the brain, as well as in neuronal cell types, particularly in the cortical neurons. And because of all this knowledge, we had the hypothesis that autosomal recessive deficiency of TMF1 underlied herpes simplex encephalitis in both patients by disrupting brain-intrinsic immunity. So to find out first, what effect did the essential splice mutation had on patient 2, we did exon trapping as well as subsequent topo cloning, and we found that there were three isoforms of TMF1 being formed by patient 2 fibroblasts, which we term M1, M2, and M3. So M1 is actually a 7-amino acid deletion in the intracellular domain near the C-terminal tail of TMF1, while M2 is the deletion of the entire part encoded by exon 10 with a addition of 48 amino acid encoded by 3' UTR, while M3 is the deletion of the entire part encoded by exon 9 and 10 with the same insertion of the 48 amino acid encoded by the 3' UTR. And through topo cloning results, we can see that majority of the isoforms of TMF1 being formed is of the M1, while M2 and M3 make up the minority proportions being expressed in the patient 2's fibroblasts. We next went on to check the localization of the different patient variant and isoforms of TMF1. And through flow cytometry with or without the permeabilization of TMF1 overexpressing hepatocyte cells, we found that patient 1 P44A variant as well as patient 2 M1 isoform were expressed in its entirety on the surface of the cell, much like wild-type TMF1, while M2 is expressed a little bit on the surface of the cell, while in its majority intracellularly, while M3 is expressed in its entirety intracellularly. And this result is again confirmed by confocal microscopy, where you can see that the TMF1 being stained in green is expressed on the surface of the cell for those HeLa cells overexpressing P44A variant as well as the M1 variant, while the M2 and M3 were expressed either majority or in its entirety intracellularly. Next, we wanted to test the function of TMF1 in the context of a viral infection, whether or not it had an antiviral function or not. So through collaboration with our dear collaborator, Dr. Greg Smith from Northwestern University, who kindly provided us with this early RFB reporter HSV1 viral replication assay, we transfected HeLa cells with the different TMF1 variants, including wild type and empty vector controls, and infected the cells with this early HSV1 viral replication assay. And we found that those cells that were expressing wild type TMF1 labeled in green over here had lesser infection of HSV1 signified by the RFB being produced during viral replication, showing that TMF1 perhaps had a little bit of antiviral effect or restricted viral entry during the very early phase of viral infection because we did the infection at 10 hours post-infection. But when we look at the cells, the HeLa cells overexpressing the different patient variants, we can see that the patient 1 P44A variant was a complete loss of function, and this is the same with patient 2 M3 isoform overexpressing HeLa cells, where we see that the RFP intensity were similar to cells overexpressing empty vector. But we can also see that the M1 and M2 isoforms from patient 2 were still functional in restricting HSV1 infection, much like wild-type TMF1 overexpressing HeLa cells. But this is overexpression data, of course. So we looked at the endogenous TMF1 function in the cell type that matter, which is cortical neurons, which happen to express the highest endogenous TMF1 level. So thanks to Gigi Notangelo from NIH, as well as Dr. Lawrence Studer, as well as Dr. Nupur from MSKCC, who differentiated the TMF1 KO iPSCs into TMF1 KO neurons. We did the same HSV1 early viral replication assay. And we found that neurons that were absent of endogenous expression of TMF1 in the TMF1 KO neurons, they had a higher percentage of neurons being infected when compared to the healthy control neurons. And they also showed a higher RFP intensity, showing that there were more virus entering the neurons as well as replicating within the neurons, showing that TMF1 had an antiviral function, perhaps restricting the very early part of viral entry or replication. We also did the same experiment with patient HPSC-derived neurons, where we took the patient fibroblasts and differentiated them into iPSC and then into patient-derived neurons. And we had the same conclusion, whereby the patient-derived cortical neurons had a higher percentage of infection versus the control derived neurons. And the RFB intensity was also higher in patient-derived cortical neurons versus the control neurons. So now that we show that TMF1 had a antiviral function, perhaps restricting HSV1 entry at the very early stage of viral infection, could we decipher the underlying mechanisms of how TMF1 function as a restriction factor against HSV1 infection? So for this, we looked at the glycoproteins and the host viral receptors that were very well published and studied that mediated the viral HSV1 viral entry into the cells during infection, which are GB, GC, GD, GH, and GL, as well as host receptors, Nectin-1, HVM, and PILR alpha. We did a simple co-IP assay, and we found that TMF1 interacted with Nectin-1, which is a major host receptor for herpes simplex virus 1. And the reverse IP is also true. Nectin-1 interacted with TMF1, but we did not see any interaction between TMF1 and HVEM, as well as TMF1 and PIRR alpha. We proceeded to do the same co-IP assay with the different glycoproteins of HSV1 that were well studied and well published to mediate the viral entry. We found that TMF1 did not interact with any of them, including GB, GC, GD, GH, and GL. So having shown the interaction between TMF1 and Nectin-1, what is actually the tripartite relationship of TMF1, Nectin-1, and Nectin-1's well-established interaction partner GD? So we had two hypotheses at this point. So first, TMF1 restricted Nectin-1 mediated entry by physically blocking the interaction between Nectin-1 and GD, hence blocking downstream viral fusion and viral entry. While hypothesis 2 is that TMF1 blocks GD and Nectin-1 activated viral fusion and the endocytosis event, but not the physical binding between Nectin-1 and GD. So to prove hypothesis 1, we did a very simple binding assay where we used a recombinant GD with a histag, and we incubated it with cells stably expressing Nectin-1 with or without endogenous expression of TMF1. And we found that with or without endogenous TMF1, the GD bound to Nectin-1 at the same level with or without the expression of TMF1. So this is unfortunate for us because it could have been a very simple explanation of physical inhibition, right? But that's not the case. So we move on to hypothesis 2, whereby we look at the internalization, which is the viral fusion, and endocytosis of the virus with or without endogenous TMF1 expression. So in the same setup, we used GD recombinant GD incubation with HEC293D cells that were KO of TMF1 or wild type with endogenous Nectin-1 expression. And we looked at the surface expression of Nectin-1 to kind of signify the internalization of the virus or GD. So in this case, if you look at the gray line, which is the wild type HEC293D cells, we can see a gradual internalization of Nectin-1, which is what happens during HSV-1 infection. But in the absence of TMF1 expression in these HEC293D cells, you can see a more intense and more aggravated Nectin-1 internalization with incubation of a recombinant GD, HSV-1 GD. And the same conclusion was also reached when we incubated with whole live HSV-1 in comparison between wild type and TMF1-KO HEC293D cells. More importantly, in the cell types that matter, the cortical neurons, we did a beta-lactamase entry assay kindly provided to us again by Professor Greg Smith from Northwestern University. And how this assay works is very simple but very beautiful. So before the start of the infection or the absorption phase, we preload the cells with CCF2 substrate. And during the virus absorption phase, we use a infectious clone with a beta-lactamase fused tegument. So during the virus entry, the tegument will be released into the cytosol and break the substrate, which releases a green in its unbroken form into a blue coumarin signal in its broken form. So in essence, with increased viral entry, we can expect a more intense blue signal and a weaker green signal. So we did the assay in cortical neurons that had a endogenous expression of TMF1 or they were KO of TMF1. And we found that neurons that were absent of TMF1 had a much weaker green signal and more intense blue signal compared to the control cortical neurons, showing that there were more intense HSV-1 entry during the first hour of a virus infection. So in conclusion to the first part of the talk, we found the first human restriction factor for HSV-1 in the brain. So in healthy neurons like yours and mine, we have a functional TMF1 that blocks the GD Nectin-1 mediated viral fusion and downstream endocytosis, and hence prevents infection in our neurons. While in the two patients with HSE, they had an absence of functional TMF1, and this led to increased GD Nectin-1 mediated entry and endocytosis, resulting in enhanced infection of HSV-1 in their neurons, and hence the clinical presentation of HSC that we see in the patients. So for the second part of the talk, I would like to shift gears and move on to another disease known as isolated SARS-CoV-2 encephalitis. Of course, many of us already know about COVID-19 and its wide-ranging clinical manifestations. Patients, or rather individuals who are infected, they can be asymptomatic on one end to being critically ill with COVID-19 pneumonia requiring intubation on the other. Well, isolated SARS-CoV-2 encephalitis is one form of life-threatening clinical presentation in SARS-CoV-2 infected individuals. And this is extremely, extremely rare and very severe and life-threatening. Well, in collaboration or in conjunction with COVID Human Genetic Effort Consortium, and also in collaboration with the Equal Lab in University of Gothenburg, as well as Charlie Rice's lab in Rockefeller University, and again, Lawrence Studer's lab in MSK-CC, we recruited 16 individuals with or patients with isolated SARS-CoV-2 encephalitis. And we looked at their whole exome or whole genome sequencing data for variations in 22 known or implicated viral encephalitis causing genes. And using the same optimized variant selection pipeline, we found a single patient with homozygous mutation in DBR1. So what is DBR1? DBR1 is the only known human RNA lariat debranching enzyme. And how it functions is during the splicing event, there is a byproduct of RNA intronic lariat that forms. And TBR1 acts by splicing the branch point of the lariat into a linear strand of RNA so that it can be degraded downstream. Well, TBR1 deficiency causing viral encephalitis, specifically brainstem viral encephalitis, is not new because in 2018, Zhang Shening and her co-authors, they already published that TBR1 deficiency can predispose individuals to brainstem viral encephalitis arising from HSV1 infection, influenza B virus infection, as well as norovirus infections. Well, in this case, we found a SARS-CoV-2 brainstem viral encephalitic patients with homozygous I120T mutation in DBR1. This boy, which we call P1, is a 14-year-old boy of Syrian descent. Through West sequencing, we found that he had a homozygous I120T mutation in DBR1. And during the encephalitis episode, he was found to have inflammation in the thalamus as well as the pons and the cerebellum following SARS-CoV-2 infection. He also had episodes of oral inflammation as shown in this picture over here. And during the encephalitis episode, he was treated with ventricular drain, steroids, IVIG, IL-1 blockade, a cocktail of antivirals, and fortunately he recovered with no neurological sequelae. And you can see from the family tree that he had three other individuals who were homozygous for I120T mutation in DBR1 as well. But fortunately, they did not develop isolated SARS-CoV-2 encephalitis, perhaps due to vaccination. Well, if we look at the DBR1 population genetics, we can see that the I120T is a private mutation. That means that no other individuals in public databases had this variation either in homozygosity or in just a single allele. And when we look at the CONNET score, this genetic variance of DBR1 is likely to be inherited in the autosomal recessive fashion. Well, if you recall, just a few minutes ago, I talked about the HSV1 brainstem viral encephalitis arising from homozygous I120T mutation in DBR1. Well, this family had the same mutation as the current patient that we are looking at, which is the I120T mutation. And through SDH analysis, we found that the patient with herpes simplex virus, brainstem virus encephalitis, and this patient with isolated SARS-CoV-2 brainstem virus encephalitis, they perhaps originated from the common ancestor almost 21 generations ago. Anyway, so next part of the study, we wanted to see again if in this patient fibroblast, is the DBR1 biochemically stable or not in its mRNA as well as protein form. So we looked at the mRNA expression on the N-terminal and C-terminal tails, and we found that for the mRNA, it is stable for the patient, as well as the previous patient with HSV1 brainstem viral encephalitis, and P1's sibling with homozygous I120T mutation in DBR1. However, when we look at the protein expression in Western blot, we can see that the protein being expressed in the patient fibroblast or individuals with homozygous I120T, they were lesser as compared to the healthy controls and the wild type or heterozygous siblings, showing that at the protein stage, DBR1 is biochemically unstable with homozygous I120T mutation. But what's more important is that when we look at the function of DBR1, when we look at the levels of RNA lariats, in this case, ID1 lariats or DKK1 lariats, we see that there is an accumulation of RNA lariats in the fibroblasts of individuals or patients with homozygous I120T mutation in DBR1 as compared to the healthy controls or siblings with homozygous wild type or heterozygous for the I120T mutation. Importantly, we are able to rescue the lariat accumulation phenotype using exogenous wild-type DBR1 transduction. So in the patient fibroblasts, when we transduce the wild-type DBR1 expressing lentivirus, we can actually reduce the lariat levels to the healthy control fibroblast intronic RNA lariat levels. Importantly, when we look at the patient derived hindbrain neurons shown here in green, we can see that these cells also had the lariat accumulation phenotype that we see in the fibroblast. And more importantly, when we perform the SARS-CoV-2 infection in these hindbrain neurons, we can see that the hindbrain neurons with a homozygous I120T mutation in DBR1 had a higher level of infection as compared to the healthy control-derived hindbrain neurons, as well as even the IFNAR1 deficient or TLR3 deficient patient HPSC-derived hindbrain neurons. To show the causality of the increased susceptibility to SARS-CoV-2 infection is due to the increase in lariat levels, we also performed this experiment in collaboration with Daxing Gao, our collaborator in China, whereby we exogenously introduce DKK1 lariats into the hindbrain neurons to see whether or not they had an increase in susceptibility to SARS-CoV-2 infection. And indeed they did. So after the transduction event, we can see an increase in DKK1 lariats being accumulated in the hindbrain neurons, in wild-type hindbrain neurons. And upon infection, we can see that those wild-type neurons being transduced with DKK1 lariat expressing lentivirus, they had a higher infection level compared to wild-type hindbrain neurons being transduced with empty vector expressing lentivirus. So follow-up study from Daxing's lab in China completed the full circle, whereby they found the underlying mechanism of how lariats affected the antiviral immunity of cells to viral infections. So through 3 papers, we showed that with DBR1 deficiency, there are accumulation of RNA, intronic RNA lariats in either fibroblasts or hindbrain neurons. And this accumulation of RNA lariats led to the impairment of the formation of stress granules, and this inhibits the activation of PKR, hence inhibiting downstream PKR-dependent antiviral pathways. So to end off the talk, I would like to give a little bit of a advertisement almost. We are now searching for patients with severe viral infections or disease, life-threatening disease arising from viral infections that are usually common or benign. And some of the patient cohorts that we're looking at are arboviral diseases like dengue or chikungunya, as well as enterovirus 71 infections with CNS complications or any case of viral encephalitis. So if you have links to these patients and if you would like to collaborate with us, please feel free to email me here. So to end off today's talk, I would like to thank, of course, members in my lab, as well as all members of the Casanova lab transatlantic in both New York as well as Paris. In particular, Dr. Jean-Laurent Casanova and Professor Shening Zhang for their supervision, as well as our dear collaborators and the funding that made the work possible, as well as most importantly, the patients their families, the clinicians, and fellow co-authors that make the discoveries possible. Thank you, and welcome any questions that you may have.
Thank you, Dr. Chan, so much. That was fascinating and so enlightening. I would invite anyone who has questions to please put them in the Q&A section, and we'll get to them as we have time. I guess I'll kick it off by asking you, Dr. Chan, what you think the main metabolic pathways that are dysregulated during viral encephalitis are, and what approaches we might utilize to treat them.
Okay, so I think through the work by Jean-Laurent Casanova as well as many other groups in the world, when we look at life-threatening viral diseases as a whole, not just for viral encephalitis, most of the time they are, deficiencies in the type 1 interferon response that gets affected, right? Because that is the main, almost the immediate response of the host against the viral infections. And this is not just in the case of genetic deficiencies, because there are also discoveries of neutralizing autoantibodies against type 1 interferons that you can imagine, right? Neutralizes the type 1 interferons against these viral infections. So, having an impairment in general of the type 1 interferon response against the viral infections can be really life-threatening for the patients or individuals who are infected.
Thank you. What areas do you see that aren't being explored in viral encephalitis that you wish there was a little bit more focus on?
I think while looking at the host systemic antiviral pathways, like for example, interferon response is pretty systemic, right? If we look at organ intrinsic or cell intrinsic immunity, I think that is really interesting because if you look at the discovery of TMF1 as a restriction factor, it is really a cell intrinsic immunity, right? It's a cell intrinsic protection against a herpes simplex virus 1 infection, which wouldn't really play a role in other parts of the body. So I think that is one area that we can put more focus on, cell intrinsic immunity against viral infections.
No, I completely agree with you. And with the new models that we have, it certainly seems like a high potential. I
Yeah.
see a question from Dr. Sten Vermun, the Chief Medical Officer for the GVN. Is there any particular viral encephalitis that you think could have genetically influenced outcomes more than others? Or do you think that most such viruses will have a common genetic risk factor?
To answer this, I think there are genetic deficiencies that affect specific viruses, like for example, TMF1 deficiency just affects HSV1. But of course, if like I mentioned just now, if you have a very important antiviral factor like your interferon response being affected, then it could be a general antiviral response shut down in the particular individual. Yeah, so there could be two aspects, either specific to a virus or general.
No, I think it's very fascinating, and there's a lot to learn about risk factors for viral infections and genetic of hosts.
Yeah.
We have another question. In your opinion, is the encephalitis producing phenotype specific to the virus, the host, or both? Some viruses have a marked encephalitis phenotype uh, based on point mutations, and in those cases, the host would have less weight versus the virus.
Yes. So I think it could be both. Um, so for this, we would like to think that for the host to be really the problem, right? Genetically, it has to be pretty rare, and by definition, actually, viral encephalitis it's extremely rare. It's a 1 in 10,000 to 20,000, right? Of infected individuals, and that is extremely rare. So for that to have a genetic cause, I think will be more highly probable versus, virus that are, neurotropic in general, right? So those could be more towards the virus side. Maybe the virus is more adapted to infect neurons or the CNS and hence causing clinical presentation in the brain. That could also be a factor where we see encephalitic cases in that case.
Thank you. And we have a question from Dr. Anders Valny. I'm sad that we can't allow people to answer live on this, so I am going to read it for you. But how phenomenal to have him for this chat. HSV-1 encephalitis is an astroglial cell infection that he showed almost 50 years ago and wants to know if you've tested the glial cell infection in this model.
No, we have not tested the glial cells model. The reason why we use the cortical neuron for this purpose of the study is because in the patients they have a 95% of the patients with herpes simplex encephalitis are forebrain and 5% are brainstem. So we use the cortical neuron model to almost represent the forebrain encephalitis. And hence, that's the reason why we didn't test it yet, the clear model.
No, but it'll be fascinating to look at. Certainly, we see lots of differences between neurons and glial cells for alphavirus and other viral encephalitis.
Yes.
We have a question from William Hall, research on CMV encephalitis in immunosuppressed patients. Have you looked at CMV encephalitis in immunosuppressed patients?
No, not at the moment. We didn't look at the CMV encephalitis cases yet.
So a lot of potential areas for collaboration, which I do encourage people to reach out to Dr. Chen to follow up after this. Yes, thank you, Dr. Anders Valny. Neuronal parenchyma do not have receptors for HSV-1. That is very helpful to know. Do we know about the... Sorry, I'm just going to answer that live. Did you look at any perturbations in like the blood-brain barrier in these specific cases?
No, we did not look at perturbations. For the case of HSC, because HSV1 is, mainly they lie dormant already in the brain, so it's not really a passing of blood-brain barrier. So we didn't look at the perturbations of the blood-brain barrier in this case.
I guess I'm curious if there are things being secreted by infected cells that would change the permissibility of the blood-brain barrier or change its impact just with the symptoms that we see associated, but perhaps with some of your multi-omics approaches where you're able to detect things that are changing between this subpopulation and another.
Yes, I think this is a very interesting point. I think technology has advanced to a point where we can use even co-cultures of different CNS cell types. So that is something that we can definitely look at in the future.
Fantastic. I don't see any other questions in the chat, and so I'd like to thank you, Dr. Chan, for a fantastic webinar on the genetics and background for viral encephalitis, and invite everyone to join us on August 28th at 11 a.m. Eastern Time, where we'll have Dr. Nadia Samaguru present on viruses without borders, global health response research in pandemic responses. That will also be continuing medical education eligible. And so we encourage you to reach out to find out how you can do the things needed for that. And again, thank you, Dr. Chan, so much. We'll be posting this later. And we encourage everybody to reach out to collaborate. And please provide any nominations for speakers you'd like to see on this webinar series.
Thank you so much.
Thanks, everyone.